1.2. Tick the box that gives the name of the stage of mitosis shown in figure 1. So in figure 1 we can see the chromosomes, we can see that the DNA has replicated and two chromatids are joined together by centromeres. So therefore interphase has already happened. They have condensed and become visible during prophase. They've not started to line up yet, so it must be prophase. So it's 1 mark for 1 box ticked at prophase. When preparing the cells for observation, the scientists place them in a solution that has a slightly higher or a less negative water potential than the cytoplasm. This did not cause the cells to burst, but it moved the chromosomes further apart in order to reduce the overlapping of the chromosomes when observed with an optical microscope. Suggest how this procedure moved the chromosomes apart. So it's the idea that water would move into the cells or move into the cytoplasm by osmosis, and this is because, as it tells us in the question, the solution has a slightly higher water potential. So water will always move from a higher water potential to a lower water potential. So in this case, it will move from the solution into the cells via osmosis. And that will then cause the cell or the cytoplasm to get bigger. They will not allow any reference to water moving into the chromosomes or the nucleus. It's just the idea that the water is moving into the cell. They will accept the idea of there being a greater volume or it's swelling or expanding for getting bigger. Ignore references to pressure or turgidity and chromosomes being more dilute, and ignore any references to changing the water content of the cell. But they will allow error carried forward for the nucleus expands, but not for the chromosome expands. So one mention of the nucleus wouldn't get you a mark for marking point 1, but you could still get a mark for the second point if you've taught about the expanding.